Hi everyone, welcome to our series of videos on macromolecules, and in this video we're going to look at lipids. Now in contrast to the other classes of molecules that we'll look at in this section, lipids are not actually polymers because they don't have repeating monomer subunits. Instead, they're all grouped together due to a single property, hydrophobicity. Regardless of structure, lipids are all lipids because they don't mix with water. There are three main important types of lipids in biology, and these are triglycerides, or fats, phospholipids, and steroids. Triglycerides are so called because they have three fatty acids, tri being the prefix for three, like triangle and tricycle, joined to a glycerol molecule. The R shown on these fatty acids here means that it's a hydrocarbon chain, meaning just carbon and hydrogen of an unspecified length. The chains can be different lengths, and they can have either single or double bonds between the carbons, which we'll discuss more in just a moment. The major functions of triglycerides in the body are long-term energy storage, insulation, and the protection of internal organs. If you consider delicate structures like your kidneys and where they're located in the body, without some amount of cushioning, they'd be banging into hard structures like bone, and would likely get bruised and damaged relatively quickly. Your internal adipose or fat tissue is one of the things that prevents that from happening. Now we mentioned that fatty acids within triglycerides can have either single or double bonds between the carbons. This is what defines a fat as being saturated or unsaturated. A saturated fat is said to be saturated with hydrogen. It contains as much hydrogen as it possibly can, due to the carbon-carbon bonds all being single. This results in straight molecules, which compact together tightly, resulting in fats that are solid at room temperature. These are usually found in animal fats like butter, although a notable exception is fish, which typically have unsaturated fats. If, however, we start introducing double bonds between carbons, then we reduce the amount of hydrogen that the molecule can contain, since the carbon can only form 4 bonds in total. This makes the molecule unsaturated. Now, because of the double bond, naturally occurring unsaturated fatty acids typically have a bend in them, and this makes it harder to pack the molecules together, making the substance less dense and a liquid at room temperature, like olive oil and other plant oils, with the exception of tropical oils like coconut. If you've looked at the back of a food packet or have used a food tracking app, you might have noticed the terms monounsaturated and polyunsaturated when listing fats. That tells you whether the fatty acids have one, mono, or more than one, poly, double bonds. You might also have noticed the term trans fat. Trans fats are a type of unsaturated fatty acid, but recall the cis and trans isomers that we discussed in the previous section. In the cis isomer, the groups were on the same side of the double bond, and this is the case with most naturally occurring unsaturated fatty acids. The hydrogens are on the same side, which causes the molecule to bend, as shown in the top example here. In a trans fatty acid, the hydrogens are on opposite sides, which makes the molecule straight like a saturated fatty acid. Trans fatty acids have historically been produced synthetically by the process of hydrogenation, adding hydrogens to unsaturated fatty acids. This was used to improve the texture and shelf life of processed foods, but it turns out that from a health standpoint, they are even worse for causing cardiovascular disease than saturated fats, to the point that the FDA actually bans them. So hopefully, next time you look at your food packet, you'll see the trans fat listed as 0 grams. The next type of lipid that we're going to consider is a phospholipid. And you may remember that we mentioned these briefly in the previous section when we were looking at the phosphate functional group. Phospholipids have a similar structure to triglycerides, but only have 2 fatty acids instead of 3. The third position on the glycerol is bonded to a phosphate group, with some other attachments added to it. And this contrast of non-polar fatty acids and charged phosphate gives phospholipids a really unique property in that they are what we call amphipathic, both hydrophilic and hydrophobic at the same time. This is important in how phospholipids function in cell membranes. When placed into an aqueous or water-based environment, like a cell, phospholipids will automatically self-assemble into a structure called a bilayer, where the hydrophilic heads will form a barrier both outside and inside the cell, facing the water and shielding the fatty acid tails in the core of the membrane. We'll come back to this in a later section when we look at the function of the plasma membrane. Our final class of lipids that we're going to look at is steroids, and these are markedly different in structure from the other two. There are no fatty acids and no glycerol. Instead, we have 4 rings of carbon all fused together as the core of the structure. You might be wondering how steroids are different from each other if they all have the same structure. Well, it comes down to the functional groups. For example, let's compare these 2 steroids, cholesterol and cortisol. The basic structure of the two is the same, but if we consider the functional groups attached here, you can see some significant differences, and these will make the molecules behave differently. Cholesterol, for an example, is a component of cell membranes, while cortisol and others like estrogen, progesterone, and testosterone, to name just a few, function as hormones to coordinate the development and function of various tissues within the body. So this is one of those cases where a small change can make a really big difference. Thanks for watching, and please make sure to check out the other videos on the other classes of macromolecules.